Let's start with the basics. What exactly are extracellular vesicles, or EVs? Their name pops up everywhere in biomedical research these days. But what sets them apart from other cell components?
Great question. Extracellular vesicles are tiny, membrane-bound particles that cells release into their surroundings. Think of them as molecular packages inside. They carry proteins, nucleic acids, and signaling molecules. What's fascinating is that EVs act as couriers, facilitating intricate communication between cells, far beyond what chemicals in blood or lymph alone can achieve.
That courier analogy is really helpful. Is their content random, or do cells select what gets packaged into EVs?
It's definitely not random. Cells sort specific cargo into EVs depending on their state and the environmental cues they receive. For example, stressed or cancerous cells might load their EVs with growth-promoting factors or genetic material that can alter nearby cells' behavior, almost like sending coded instructions to shape their surroundings.
So, EVs are more than passive debris. They're active players in cellular strategy. Before we get into disease implications, can you give an example of EVs in action within healthy tissues?
Absolutely. In the immune system, EVs help coordinate responses by ferrying antigens and regulatory molecules between immune cells. They ensure swift information transfer, much like secure email between key staff in a company. This fine-tuned communication is crucial for mounting appropriate defenses and maintaining tissue health.
Building on that, how do EVs function in cancer? Specifically, what makes tumor-derived EVs such a hot topic in oncology research?
Tumor-derived EVs are pivotal because they carry instructions that can remodel their environment in favor of the cancer. For instance, they can enhance tumor cell invasion, help prepare distant organs for metastasis, or suppress immune responses. In a sense, they're like advanced teams, scouting and conditioning new territory for cancer's spread.
That's striking. Are there particular molecules or signals within these EVs that drive this process?
Yes. These vesicles often contain microRNAs, proteins, and even metabolites that can rewire the gene expression and metabolism of recipient cells. A 2018 study in Nature Review's Clinical Oncology highlighted how EVs carrying certain microRNAs set up pre-metastatic niches microenvironments in distant organs that become more receptive to cancer cells, almost like planting seeds in fertilized soil.
Are there differences in how EVs interact with cancerous versus non-cancerous cells?
Absolutely. Tumor EVs can manipulate non-cancerous cells, such as fibroblasts or endothelial cells, nudging them to support tumor growth and invasion. For example, they can induce fibroblasts to become cancer-associated fibroblasts, which then remodel tissue architecture, promote blood vessel formation, and dampen anti-tumor immunity. This web of influence makes targeting EVs an exciting therapeutic prospect.
Let's pivot to a more physical perspective. What role do mechanical forces play in EV biology, especially within tumors?
The intersection of mechanics and cellular signaling mechanobiology is transforming our understanding of cancer. Tumor tissues are often stiffer due to changes in their extracellular matrix. This stiffness doesn't just affect how cells move, it directly influences EV production and targeting. For example, studies show that when the matrix is rigid, cancer cells ramp up EV secretion and alter their cargo, enhancing their ability to promote metastasis.
That's fascinating. Could you use an analogy to help visualize this process?
Certainly. Imagine EVs as delivery drones. In a dense urban environment, drones need to navigate differently than in open fields. Similarly, in a stiff tumor matrix, EVs are released with cargo tailored for that challenging landscape, helping cancer cells break through barriers and colonize new areas. Mechanical cues essentially recalibrate the EV's mission parameters.
Are there real-world examples where manipulating tissue stiffness changes cancer progression via EVs?
Yes. For instance, a 2021 biomaterials study showed that making the extracellular matrix softer reduced exosome release and slowed breast cancer cell motility. Conversely, stiffening the matrix increased EV-mediated signaling, accelerating tumor growth and invasiveness. This suggests that therapies targeting tissue mechanics could indirectly disrupt EV-mediated cancer spread.
To round out our discussion, how do EVs contribute to the complex interactions between cancer cells, stromal cells, and immune cells within the tumor microenvironment?
EVs are crucial mediators in this cellular conversation. They help cancer cells recruit and reprogram stromal cells like fibroblasts, making them more supportive of tumor growth. Additionally, EVs modulate immune responses, sometimes suppressing the activity of cytotoxic T cells or skewing macrophages toward a tumor-promoting phenotype. This dynamic crosstalk essentially builds a protective niche around the tumor.
It almost sounds like EVs are diplomatic envoys, brokering alliances in the tumor's favor. What are some implications for therapy?
Exactly. By targeting or engineering EVs, researchers are exploring ways to disrupt these harmful alliances. For instance, blocking EV-mediated delivery of immunosuppressive signals may enhance cancer immunotherapies. Alternatively, engineered EVs could deliver drugs or gene editing tools directly to tumor or stromal cells. The challenge lies in achieving specificity and minimizing unintended effects on healthy tissues.
Given the complexity and adaptability of these networks, do you foresee EV-based diagnostics or treatments entering mainstream oncology soon?
The field is moving rapidly. Liquid biopsies analyzing EVs from blood or other fluids are already being tested for early cancer detection and monitoring therapeutic response. As for treatments, clinical trials are underway, though hurdles like large-scale EV production and targeting remain. Still, the potential is immense. EVs could become both the black box and the toolkit of next-generation cancer care.